Thank you very much, Naomi, for organizing that. Thank you very much, Sook Lei, to come from the remote Los Angeles in a few seconds. That's quite fantastic. So we tried to invite Sook Lei 1 year ago, or at least 1 year ago. And so the plan was that coming this year, it would be no problems that she's coming in real. But it will be for another year. I think they will. So airlines will be overcrowded in a few years. We will have to catch up all this meeting, real person meeting. So Sook Lei is working at USC, so University of Southern California in Los Angeles. I had the pleasure to visit. So they have a very important center for rehabilitation, research in neurorehabilitation. I don't know when I visited. That was 2 years or 3 years ago. Because
few years.
I lose track with time, especially now. So anyway, so Sook Lei is working in neurorehabilitation, especially she has a background in from what I understand, in occupational therapy, kinesiology and occupational therapy. She did a PhD at USC 2012, and after she was at NIH for how many years?
2 years.
2 years, and after you were directly back at USC as assistant professor?
That's right. Yeah. It was enough cold weather for me.
So when I was visiting USC, I was very interested to discuss with Sook Lei, and so I would like to continue discussing, but first we have to learn more about what she's doing. So thank you very much for coming, and I look very much forward to your talk.
Thank you so much. Thank you everyone for having me today. It's a real honor to be able to speak with you. I'm going to go ahead and share my screen. Let's see. Can you see my slides? Yes? Are you able to see my slides?
Yes, that's perfect.
Okay, perfect. Okay, great. So today I'm going to talk about big data brain imaging, virtual reality, and neurofeedback for stroke rehabilitation. It's kind of like a high-level overview of the types of research that my lab does. Hopefully by the end of this talk, you'll be able to have a better understanding of how we use big data brain imaging to provide more robust and specific insights into how people recover after stroke. And you'll also be able to identify some ways that we can use virtual reality with brain and muscle computer interfaces for more personalized stroke rehabilitation. So to start out with, what you see here is blood flow to the brain in a real person. It's actually in my husband's head. And one of the great things about this is that it visualizes what happens on a second-by-second basis for most of us. However, when somebody has a stroke, blood flow to the brain, like what you see here, gets cut off. And if that happens for more than about a minute or two, then brain tissue can actually start to die. And this leaves people with long-lasting sensorimotor and cognitive and all sorts of behavioral deficits. So there's a few sad facts about stroke rehabilitation that really have been driving the work that I've been doing. So one is that over the last 20 years, over 90% of our stroke rehabilitation clinical trials have failed. And what this means is that while we try to come up with better and better technologies and treatments for people who after stroke to be able to recover, a lot of them are not doing any better than our usual care. So we're not really coming up with new therapies, especially for people who might need it most. In addition, one thing that we're constantly learning and seeing is that stroke recovery is highly variable. So when we get a new patient for treatment, it's not one size fits all. We can't do the same thing with each person. It's highly individualized, and that individualization is based on a number of factors that I'll talk about in a bit. So because of that, in my lab, we've been working really hard to change the question from does this treatment work to who does this treatment work for, and trying to move towards the idea of precision rehabilitation. So one of the best biomarkers or indicators of stroke recovery is brain imaging so far. And with brain imaging, one of the things we particularly focus on is the volume of the lesion and also the location of the lesion, more importantly. So in these three brains, these are three different individuals with stroke. hopefully you can identify at least one or two of the lesions. So in this brain, the lesion is here. In this brain, the lesion is here. So the dark center represents tissue that's dead and gone after the stroke. And in this person, they have a relatively small lesion, but it's right here. The interesting thing is that for all of three of these people, they have the same amount of sensorimotor impairment after stroke, which means they all have the same amount of difficulty moving their arm after stroke. And it shows us that it's not just the volume of the lesion, but it's also the location. So critically, these lesions all fall within what's called the cortical spinal tract, which is the descending motor pathway from the cortex, from the primary motor cortex, down to the muscles of the hand. And because of that, they have similar amounts of impairment. So ideally, what we would be able to do is take a new stroke patient and compare their brain and lesion location and also the integrity of the rest of the tissue of their brain with thousands of other brains. But in order to do that, we would need obviously thousands of brains and a huge database of not only the brains, but also the associated behavior. What we would ideally like to be able to do is say, for any given new brain, they look the most like these other cluster of individuals that we've seen before, and these cluster of individuals had this prognosis, this type of recovery, and responded best to this type of treatment. So this type of learning and algorithm is something that's similar to what Netflix does when you watch a movie and you watch one and then they compare your preferences and likes to millions of other people that they have in databases and can suggest for you what you might like based on your own history and based on the history of millions of other people. However, getting thousands of brains like this is much harder than I think Netflix has a time in terms of getting data for them. For us, we need to bring in patients, we need to put them in the MRI scanner. This is both costly and time consuming. And a lot of people after stroke don't really want to be a part of a research study because they just want to get back to their normal lives. So one way that we've approached this is through joining forces with ENIGMA. ENIGMA is a consortium led by my colleague, Dr. Paul Thompson. The goal of ENIGMA is to collaborate to solve problems that no one group could solve alone. And basically what that means is we meta-analyze and mega-analyze data collected around the world by pooling our data together across research groups. So I am the chair of the ENIGMA Stroke Recovery Collaboration. In our group, we currently have over 100 researchers representing, actually at this point, it's over 50 research cohorts worldwide. We have over 3,000 stroke MRIs with accompanying behavioral data sets, and we continue to grow on a daily basis, which is great. And so this is our attempt, since no one group, I think for one group to collect 3,000 stroke MRIs in behavior, especially in a research setting, so I'm not talking about clinical hospitals who are getting this type of throughput, but high resolution research grade scans, I think it would take many, many years. So it kind of expedites this process and allows us to combine all the work that we're doing together. In a nutshell, how ENIGMA works is typically people will collect MRI or genetic data for their own studies. So let's say each group collects about 20 or 30 subjects for their own study to ask a specific research question. They can analyze that data, publish it, do everything that's normal in the research process. But then afterwards, they can also contribute that data to an ENIGMA analysis, which we consider like a reuse or recycle model. So once you finish publishing your data, you don't usually touch it anymore. It just sits there on a drive somewhere. So you can share it with us. And in exchange, you become an author on what is usually a very large multi-author publication. Data analysis is probably the most important part. So we make sure to harmonize all of our analyses across sites so that we can run them on all different types of data that we're getting in. This slide is showing you an author list example of one of the earlier ENIGMA papers that came out in 2015 in Nature. This paper had 283 authors, I think, but together all of those authors were able to pool their data and they had genetics and high resolution MRI data for over 30,000 people. So it just kind of speaks to the power of this type of collaborative approach. In ENIGMA, we now have over 30 working groups studying 18 different diseases, and there's more groups that get started on a regular basis as well. Briefly, some of the methods that we use. So for stroke recovery, and especially for stroke MRI and kind of harmonizing that type of imaging data, One of the biggest challenges we have is the lesion volume. That means that for us, a lot of the automated tools that we use to analyze the data are a little bit more susceptible to imperfections. So we have a whole pipeline with a lot of manual quality control and data scrubbing steps that are involved. I'm just showing a few members of my research team here because they work really hard to receive new data, sift through it, convert it into like a standardized format. We run FreeSurfer to extract subcortical and cortical volumes throughout the whole brain. And then we manually quality control all of these volumes to make sure that the segmentations are correct. We also have been working very hard to do semi-automated lesions segmentation. And I'll talk about that a little bit later. But for us, because we kind of have an idea that the lesion should be somewhat related to the brain and behavior relationships we see, we are trying very hard to be able to segment those lesions on a large scale. So one of the recent papers that we did put out a paper last year in 2020 that kind of described all of the methods that we've developed to be able to harmonize and analyze big neuroimaging data sets that are multi-site. This past year, we were able to put out a paper looking at just the volumes of subcortical nuclei that are not affected by the lesion, and to see whether this kind of residual motor architecture relates to behavior as well. So for this study, we had 828 individuals with stroke coming from 28 different research cohorts around the world. We had high resolution, so that's about 1 millimeter isotropic voxels, T1 weighted anatomical MRIs, and we use those to quantify the volumes of several subcortical nuclei. So it includes the thalamus, the caudate, putamen, the pallidum, the nucleus accumbens, as well as the volume of the lateral ventricle, which we used as a proxy for general atrophy. So essentially, the larger the lateral ventricle is, the more atrophy we would expect to see across the whole hemisphere of that brain. We then did regression for each subcortical volume with sensorimotor outcomes. So we're looking at whether sensorimotor outcomes is a significant predictor of subcortical volume. And we were able, because of the large data set, to look at several different subgroups. So we looked across the whole time course of stroke. We also looked at just people with subacute stroke. So that's within the first 90 days of stroke versus people with chronic stroke. So in that case, we used a definition of more than 6 months post-stroke to see if there are different relationships that kind of pop out during those different time periods, which you would expect because there's different neuroplastic processes that are occurring during those times. Some of the things that we found briefly are just that in early subacute stroke, only the thalamus, the volume of the ipsilesional thalamus, corresponds to sensorimotor outcomes. So that is, the larger the ipsilesional thalamus is, the better the outcome is for that person. And that's important not only because of the possible role of the uninjured thalamus after stroke, but also because we found that relationship even within the first 21 days after stroke, which suggests to us that maybe the thalamus has some sort of neuroprotective effect. So the volume of the thalamus before it's injured is also important to be able to kind of kickstart a lot of the processes for metabolism and cellular repair that are important for recovery. At the chronic stage of stroke, the thalamus is actually no longer significant. And what we find are significant relationships between the putamen, which is an important part of cortical basal ganglia loops in terms of motor control. And we also find the nucleus accumbens. So that is a region associated with reward and motivation. In both of those cases, the larger the volume of these regions is, the better the outcome is. On the other hand, the ipsilesional lateral ventricle, which is shown here in blue, suggests that the bigger the ventricle is, or the worse the atrophy on the ipsilesional side, the worse the overall outcomes. And then we finally also looked at people at sensorimotor outcomes in terms of impairment. So that's like kind of how well you can actually move your arm versus measures of function. And functions would be like how well you can use whatever amount of motor control you do have. So can you flip a card? Can you stack a checker? Things like that. And what we found is only relationships between these subcortical nuclei with impairment, which I think could be expected because impairment is more of a direct motor control issue, and these regions are more associated with direct motor control. And in particular, we found similar relationships to the chronic strokes. So the larger the putamen is, the better the outcomes, and the larger the ventricles is, the worse the outcomes. So we are basically just getting started with a lot of these analyses. That was one of our first papers, but moving forward, we have a lot more things that we're looking at. So we're looking also at other uninjured brain volumes associated more with cognitive behavior. Like I mentioned, we're getting ready to look at the impact of the lesion volume and the location of that lesion on both sensorimotor and cognitive outcomes. Another thing we can do with this large data set is look at the replicability of predictors that have been identified in smaller studies using this large and diverse data set. So the nice thing about this data set is it spans all levels of impairment, all time points after stroke, and all types of people. So I think that diversity really helps to understand whether certain predictors are really generalizable to anybody with stroke versus just certain types of people. And finally, I think one of the most important things that we've done is establish a reliable infrastructure for stroke data sharing so that we can do large-scale analyses moving forward. So at this point in our group, we have a number of people that have proposed other analyses that they're interested in using pooled data from people across the group. So things like multimodal imaging, looking at diffusion-weighted imaging or resting state fMRI, EEG, etc. One thing that I had mentioned a few times now is the need to be able to identify the lesion on a large scale for each brain. And so usually what we do in the lab is for each brain, we have to go through slice by slice, and in a normal high resolution scan, that can be over 100 or 150 slices. And we just manually outline the lesion using a tool such as MRI-Cron or some other tool to create a lesion mask. As you can imagine, this is a horribly tedious, time-consuming process, and it also requires knowledge of neuroanatomy. So it requires some expertise, and it is still somewhat subjective. So we've been trying really hard to understand how can we scale this up for thousands of brains, because even just to do 300 brains, for instance, can take about a year for a team of researchers to do well. So one of the first things that we did is we actually manually traced 300 brains, but we released it as a public data set so that it could be used to encourage better machine learning algorithms for this process. So what you're seeing here is the overlap of those 300 brains, and this data set is called Atlas. It's widely available. You can just download it from the internet, and anyone can use it. So our goal really was that by releasing this data set, because it's the largest stroke T1 weighted MRI data set with manually segmented lesion masks, meaning that it has some gold standards to it, or ground truth, that people would be interested in looking at lesion segmentation because of it. And in fact, it has turned out to be pretty widely used, and we believe that it is actually hopefully helping move the field forward to having more accurate lesion segmentation algorithms. For anyone who might be working on lesion segmentation algorithms, we welcome you to contact us if you have any questions, and also to use this data set because it's meant to provide a large, diverse training and testing data set. What we found before we did this was that most algorithms were trained and tested on smaller data sets that people had access to, so maybe 30 to 60 subjects that they had at their own research sites. So we hope that by providing this data set, it can improve the machine learning algorithms that are being developed for lesion segmentation. Another thing that it does is provide a standardized data set for systematic evaluation of how existing algorithms perform. So we actually used it to do that and compared them and found which publicly available algorithm was the best performing. Over the last 2 years, which is roughly when it was released, it's had over 1,200 downloads from lots of countries around the world, which is really fun to see. Like I said, although the purpose was to spur, hopefully, the creation of new algorithms, there's also been a lot of exciting other uses, which I just want to touch on in terms of open science and sharing data, which is that I think especially this past year because of COVID and people not being able to collect their own data, We've had a lot of requests for the data set to be used as part of thesis projects. Coursework, people use it for abstracts and manuscripts, but it provides a data set that people can use and openly access in order to kind of get started in this area. As probably many of you are familiar, deep learning is kind of the new thing and something that's been growing quite rapidly. And with the release of this larger data set, even though it's still not very large, it's large enough that people have started to use deep learning methods on it. So there's tons of papers that are coming out using Atlas to try out new network algorithms in order to segment the lesion. One of the things that we found interesting about this, though, is that a lot of people will publish their papers about lesion segmentation, but they won't share the algorithm, which makes it a little bit hard to evaluate the actual performance. And also it makes it a little bit harder to kind of move things forward. Because of this, we've collaborated with the Paris Sook Lei Center for Data Science. So we are putting together a web-based challenge so that people can actually work together and collaborate on trying to find the best method for lesion segmentation. As part of this, we'll be releasing another, I think, approximately 300 brains, and we'll actually also be having a separate data set for testing that's hidden of about 200 MRIs. So that's in addition to what we've released in Atlas already. So if you're interested in this, I hope you'll stay tuned. We'll like to share it. It's also a great tool for classes. It does have a starting kit, which I really appreciate, so that students, if they want to learn about lesion segmentation or machine learning, they can join this challenge, and it has kind of a way to get started with the data as well. So it's hopefully going to be a nice educational tool. That said, even if we were to have a great performing lesion segmentation algorithm, one of the things we still have to do is manually check the quality of the lesion outputs and make sure that they look correct. So one of the things that my PhD student, Carrie Ito, has done is create a toolbox called PILES, or Pipeline for Analyzing Lesions After Stroke. This is also freely available software on our GitHub. But basically what it will do is overlay the lesion volume or the lesion mask that's generated with whatever the brain you're looking at is. And that way you can quickly go through, and it creates a web page, and you can click like good, maybe, or no. And it will sort the data so that if the data is incorrect, we can go back and manually fix it. And if it's good, we can pass it and move it on. However, this step is still quite manual and takes time. So the next question we had is, can we scale this process up for thousands of brains as well, the quality control process? And here we took I think a little bit more fun approach. So we found inspiration for this problem in the form of Tinder, which, if anyone knows, Tinder is a dating app in which it takes a really complex problem of, do you want to date somebody or not, which really has a lot of variables that one should consider ideally. But it boils it down to a simple swiping motion. So you just swipe right if you want to date them and left if you don't. And it kind of makes a complex problem simple and quick. So we created something similar with my colleague, Dr. Anisha Keshavan, called Brander lists. So Brander was her original creation, which was to do quality control of MRI image quality. And then we added the less for lesions, where now you take a lesion volume, overlay it on the brain, and it blinks as a GIF. And you swipe right if you think that the lesion mask is correct, and swipe left if you think it's incorrect. And in this way, we hope that we've made quality control a little bit faster, but more importantly, a little bit more fun and accessible to citizen scientists. So that would be anyone in the general public who wants to get involved in science, because we feel like a task like this, while it requires a little bit of anatomical expertise, some of the things, like some of the masks that you see here, are obviously wrong or obviously pretty right. And so by recruiting people who are just interested in science in general, it could be a way to start to streamline this process and sort out which lesions we need to fix and which ones are good to move forward. With Brainderless, it has been gamified, so there is a leaderboard. So when you log in, and you can try it now if you're interested at brainderless.us. There is a player username. You get a rank based on how many swipes you've done. And you can also unlock levels based on how many swipes you've done. So for instance, at 10,001 swipes, you could become a unicorn. So hopefully it's just a fun way to kind of engage the public in scientific tasks that are a little bit more simple and approachable for them. If you're interested in this general approach, though, you could also check out Anisha's website at swipesforscience.com. So after working with us and creating Brander herself, she's also created a general platform so that you could use any type of data for any type of simple binary decision. So for instance, I think there's also one called Whaleder now, where you listen to audio clips of underwater noises. And if you think that there is a whale sound within the audio clip, you swipe right. And if not, you swipe left. So there's lots of different opportunities. It's an open platform. It's on GitHub that you can just use it and kind of modify it for what you need. Okay, so hopefully in this part of the talk, I gave you a little bit of a feel of how we are trying to use different neuroinformatics tools to combine big data brain imaging for stroke specifically. However, if you're interested in any other disorder, there are, like I said, over 30 ENIGMA working groups that focus on other clinical disorders. So things like depression, PTSD, bipolar disorder, and much more. And this is just a thank you to our ENIGMA Stroke Recovery members, of which there are many, and who have all been gracious to contribute their data in this collaborative way. In the second part of the talk, I'm going to move a little bit more to the therapy side and rehab side. So essentially in the first part, what we're trying to do on that arm of my lab is figure out better personalized neural targets based on somebody's baseline presentation, essentially. But at the same time, in tandem, we're also working on, if we knew personalized neural targets, how could we actually implement that in therapy? One way that we've been really interested in, mostly I think because it's fun and interesting, but also because there's a lot of potential, I think, is by incorporating virtual reality into therapy. So what you see on the left here is called the Sword of Damocles. This was one of the first virtual reality headsets, I think around the 1950s. He has a goggle system on, but what's interesting to me is that the goggle system is actually suspended from the ceiling and then connected to this, which is not a bookshelf, it is a computer. So this whole thing was the virtual reality, the first what we call head-mounted display virtual reality system. Fast forward to 2015, this is the Oculus Rift, which I'm sure many of you have seen, if not tried yourselves at this point. And so the Oculus is a untethered, now it's untethered system for only a few $100s, whereas this system was millions of dollars. And you can put people in head-mounted virtual reality, and they actually feel like they're immersed in a completely new world. So there's been a lot of talk and really a lot of research on using games for stroke rehabilitation because it is a fun and motivating way to encourage movement. And we know that one of the best things in rehabilitation is to encourage high doses of movement if we can. My colleague Steve Kramer recently performed a study showing that telerehabilitation with a game-based system at home, which is what this participant is doing, is actually as efficacious as in-person therapy over the course of 28 days. And so that's really important, I think, because it opens the door for these types of virtual or game-based platforms to provide therapy. And it makes it much more accessible for patients who can't come into the clinic or who don't live near a good rehab hospital, for instance. With VR, one of the things that we can do here, and I'm just going to turn this down, one of the things that we can do is not only provide a game-based system, but we can also track movements. We can track kinematics. So there's a camera on the front of our VR system to track the person's hands. And we can also control the entire environment. So this allows us to personalize the therapy just a little bit more so that if somebody has neglect, for instance, and they don't attend to their left side very regularly, then we can have the objects only appear on the left side or in the midline and shift to the left side so that it encourages more attention to that side, for instance. We can also change the amount of background contrast. We can essentially modify anything in the environment, which is something that's actually really hard to do in a normal clinical setting or even in a home setting. So this is something that we've been working on with my colleague, Dr. Skip Rizzo, and we've been creating more and more games and actually started to bring it into the clinical environment for patients to try as a general platform. However, for people who can't move their arm after stroke at all, rehab treatments are very limited. So what you saw in the previous video was my PhD student who was trying out one of the games. But as you can see, he's able to move. So what the system does is kind of just encourage more movement or exercise, which is similar to what some of the other tele-rehab systems can do. However, after a stroke, some people have moderate to severe disabilities and they can't move at all. And in that case, what we can do with them, whether they're in the clinic or at home, is really limited. One of the more promising avenues that's been explored is brain-computer interfaces to teach people to directly control their own brain or muscle activity and thereby improve control of their affected arm. And so this is an area that we've been really interested in because I think going back to the original question of how can we personalize treatment to specific neural targets, this is one way that we can do that. So this is a video that comes from the NIH when I was a postdoc there. And one of the first things that we tried for stroke neurofeedback is using MRI. So they are in a 3 Tesla MRI scanner. And we tell people to try to learn to control their brain activity coming from motor regions, which should theoretically improve motor control over time. And the way that we do that is what you see here is what the patient sees in the scanner. But they have a circle with crosshairs, and when they see that, they're instructed just to rest. And then we say, when this turns into a triangle, a red triangle pointing up, we want you to try to make this bar or thermometer bar increase. And the way that you can do that is by imagining movement, and that will help to increase activity in these motor regions where we're getting the signal from. So what you can see here is after a few runs, when the triangle goes up, it takes a few seconds because of the hemodynamic delay. But over time, people are able to control their brain activity. And this is actually looking at the brain connectivity between motor regions, 2 motor regions in the brain. And outside in the scanner, we can see that the 2 regions that we're recording from, in fact, do co-fluctuate and increase in accordance with the stimuli over time. So that's an example of real-time fMRI neurofeedback, which is a type of brain-computer interface. We studied this for a few years and found that both people who are healthy and people with stroke can learn to control their own brain activity, which is important, I think, for overall recovery. But the system itself had a few limitations. So the first is that obviously MRI equipment is really expensive and not widely available. So while it was really neat and people really enjoyed being able to have some agency over their own brain activity, even if they couldn't move their own body, it was a pretty big hassle for them to come into the MRI centers to do these studies. In addition, as you may have noticed, the feedback is relatively boring. It is just a thermometer that goes up and down. And that was actually from about 2012 or 2014. So I could have said it was from 1980s and you probably would have believed me as well. So there are some ways that we could make the feedback much more interesting, I think. And then finally, to control motor regions in the brain, participants usually had to close their eyes to imagine movement so that they could actually increase the activity. And then they would open their eyes every so often just to see if the bar had actually moved, to see if they did the task right. So this was actually the most problematic issue to us because it became more of a dual task paradigm where they're trying to do 2 things at once versus just something natural and intuitive. So moving forward, we wanted to make sure we could create something that was both portable and accessible, that was engaging to the participant, and also with feedback that would naturally activate sensorimotor regions in the brain so that they didn't have to close their eyes to imagine movement, but they could actually see on the screen, maybe, for instance, their virtual arm moving. So to do these things, we moved from MRI to EEG. For those who are familiar, EEG is also a non-invasive way of recording brain activity across the scalp. And then we also changed from a regular screen to head-mounted virtual reality. And while I've talked a little about virtual reality and how we can control the whole environment, there is another reason we chose VR for this project, and that is the fact that it's extremely immersive. So for instance, if you are, like this gentleman is, in VR and riding a virtual roller coaster, which you can see on the screen here, you actually feel like you're on a roller coaster, and you can look all around you and you can see the drop-offs, and it gets very exciting. We tried this earlier. I'm not sure if my sound will play, but if not, I will just tell you what happens. So the guy is going up on the roller coaster, and as he crests over the top, his friend pushes him His friend pushes him right as he crests over the top, which if you're on a roller coaster, that's probably your biggest fear, is that somehow the car gets loose and you start falling through space. So in this video, the guy starts falling and screaming really loudly because he feels like he has been flying through space for some time. And actually, he yells and flails for about a minute before somebody thinks to just remove the VR headset from his head, and then he's fine again. So if you weren't able to hear that, I can send you the video because it's a very entertaining one. Or you can just look on YouTube for a roller coaster VR prank because it really took off after this, I think. But the general idea and the reason I like this video is to illustrate that in head-mounted immersive VR, we can feel very embodied. We feel like we're there, and that has not only visual consequences, but also whole body behavioral and physiological consequences. So for instance, if you put on a VR headset, even though you know you're in your home or your school, just on a flat ground, if you look down and see your body standing at the edge of a skyscraper, you might actually start to get butterflies in your stomach and your palms might get sweaty. And if someone says take 2 steps forward, you might say no, because even though you cognitively know that you're at home, the visual illusion that's provided by virtual reality is so strong that it can actually change your behavior. And there are a number of different studies that have shown this to be true. So for instance, they've shown that if you give somebody in VR a child's body, they start to exhibit more childlike behaviors. If you give them an extra long right arm versus left arm, then they actually interact with the world as though they have a very long right arm, and they adapt very quickly to this. And there's actually an after effect. So even after you take off the headset, it takes a few minutes before they recalibrate their arm to its normal length again. And there's also research showing that if you give somebody the avatar of a more attractive person in VR, they start to exhibit more the behaviors of more attractive people, which are apparently speaking very close to other people and sharing more personal information, which I found very interesting. In any case, we took all this literature and thought to ourselves, can we give somebody with motor impairments after stroke the ability to control a healthy avatar in VR? And will this essentially trick the brain into thinking that it has healthy motor pathways and reinforce those brain to muscle pathways to encourage motor recovery? To do this, we created what we call REINVENT. It's basically a system that can take brain and or muscle signals that indicate that somebody's trying to move, even if they can't actually move in the real world. And it gives them feedback of their virtual arm moving. So this is considered closed loop neurofeedback, and going back to that dual paradigm issue that I talked about before, it hopefully provides a more natural way of rewarding and incentivizing people to try to move. This is an example of an earlier system we had. So in this case, this person has had a stroke. As you can see, they can't really move their affected arm, but we are recording signals from their brain. And so when their brain indicates they're trying to move, then the virtual hand on screen, which is also shown here, starts to move as well. For some people who have more motor control, they actually don't even need the EEG headset. We can just use EMG electrodes recording from their arm. And basically, if their arm muscle indicates that they're trying to move or extend in this case, then we control the virtual movement of the virtual arm based on that. And you can see also the signals of the EMG at the same time. So in both cases, we've been studying this for a few years now. Whether we give EEG feedback or EMG feedback, people are able to learn to control the feedback, and it tends to result in more concentrated brain activity over sensorimotor regions of the brain. And when we give EMG feedback, which we thought might be more effective if people had enough trace EMG activity, then we actually see increased cortical muscular coherence on the affected side, which we were pretty excited about. In addition, over the course of like 10 to 12 sessions, 6 out of 8 individuals were able to improve their motor control as measured by clinical outcome measures. However, I think one of the things we find is that there's still quite a lot of variability in how people respond to this and whether it works for people or not. And one of the things that we think is that it requires a higher dose than just 10 to 12 sessions in the lab, especially because the population we had worked with so far has chronic stroke, so it's been quite a while. So next steps, we've created what we call tele-REINVENT. So this is home therapy with tele-rehab with a clinician. And in addition, they get a laptop we're just using the EMG because it's something people can put on themselves at home. And they can see their own muscle activity, and they also play a series of kind of fun arcade games that try to emphasize and are using neurofeedback from the muscles. So this is kind of where we're going next. You can see an example of it. So this is the patient's hand with the EMG electrodes, and it drives the movement of the virtual hand. This is a game of skee ball played in a rather unconventional way because we are trying to encourage wrist extension in this case, which is something that's particularly challenging for this patient. But what we're hoping to do next is to be able to use this at home, but also do pre and post clinical measures in the lab so that we can look at neural changes and as well as neural baseline presentation to be able to understand how to best personalize therapy for each individual. Finally, this is just one of many different projects that we have going on at USC that use virtual reality. Because of this, we created what's called the USC Smart VR Center. We encourage you, if you're interested in virtual reality or augmented reality or mixed reality, any version of that, to join us. We have a bunch of researchers, educators, industry leaders, clinicians, and artists that are all working together at this center to collaborate and make VR more scientifically rigorous and clinically impactful. So with that, I'll conclude. I'd like to thank my lab. A huge thanks to all of our participants. And thank you to all of you for listening. All right, I'm gonna stop sharing, I think.
Thank you so much. There is, I think, one question in the chat. We can have a look.
Sure.
So there is really a large repertoire of works that you showed us. So, okay, Joao is asking, yeah, if there's any approach for using unsupervised learning on MRI lesion. Yeah, so I guess you have something about that.
Yeah, sure. Yeah, so there are approaches that use unsupervised learning. They tend not to do quite as well, just because there is a lot of prior information in the brain that's important for guiding lesion segmentation versus finding holes in the brain in general. So, for instance, like to detect the difference between ventricles versus lesion or normal levels of atrophy versus a stroke lesion is a little bit more complicated. But with our data set, you're welcome to try it if you'd like to. I think at this point, people are trying anything. So there's still quite a bit of room for improvement there.
Okay. Balint is asking which kind of digital tool and engine do you use to create the VR environment? The VR.
Yes. Yeah, so we mostly use Unity in our lab. And in order to pipe in the EEG and EMG information, we use Lab Streaming Layer, or LSL, integrated into Unity.
What kind of background do the people have in your group?
Yes, yeah, that's probably the most fun part. Is that, for instance, on REINVENT, we have one neuroscientist, a biomedical engineer, a VR programmer, and a occupational therapist. So they're all students of some sort or trainees, but they all have to work together quite closely to make the product in the end.
Okay. So Megan is asking, what about patients who do not respond well in the VR environment?
Yeah. Yes, so we actually tested for nausea and motion sickness early on. Luckily, in this case, it's not a roller coaster. It's just moving an arm. So most people didn't. Actually, all the people we tested didn't have any changes in terms of what we call simulator sickness. So that's like the nausea and the motion sickness and discomfort. If anything, the VR headset sometimes gets hot and sweaty inside, especially if people are trying really hard. But then we just let them take it off and take a break if that's the case. But I think nausea and motion sickness are much more of an issue if you're trying to walk or move in VR, or if you're showing somebody moving, like on a roller coaster or flight simulator or something like that.
I have a question about VR because so we have always been thinking that actually, so if I see my father who would be at the age of he doesn't have stroke, but he could from the age, and then I wouldn't see him with goggle. So that's why actually, so we created this table where you have objects and so on because we thought. Yeah, they don't like VR glasses. So what about that? We are completely wrong. How is that?
It's a great question. It's actually one of the first questions we asked, and I'll send you a video later. But initially, when we started with VR, we tested it at nursing homes. So we took it to people that are 90 and up and had them try VR, and just to see what they thought of it. It just depends on the person. So we had a woman who was about to turn 100 use it, and she loved it. She thought it was so neat to see. For her, we put her in some sort of like simulation of Tuscany, and she just liked to see butterflies and birds and flowers and things like that. Actually, the bigger issue is the head control and the neck control because the VR systems are a little heavy. So most of them actually hold the VR system with their hands because it's a little bit, in the past at least, it was too heavy for them. The other problem is that because they can't see their hands anymore, they sometimes have some difficulty to manipulate objects. So if you want them in VR and to use a joystick, for instance, or a trackpad, it's a little bit more complicated. So I think in your case, like a flat plane where their vision is not occluded is equally good, and most people will probably prefer that for simplicity. It just depends on the person.
With augmented reality glasses at right now, so HoloLens and so on.
Yes.
Is that different, or how can you compare?
Yeah, I think HoloLens will be, or like AR glasses will be a lot easier to use with patients, just because you can see what they see at the same time, and you can point at things, for instance, that make it easier to interact with them. I just think in terms of the technology, we haven't. It's not as widely available as VR is, and it's much more expensive, and the fidelity of the AR is still a little bit up in the air for us. So we haven't made that shift yet.
Okay. So Pablo is asking about what about using NIRS rather than EEG?
Yes. So we haven't used FNIRS directly. Some of our colleagues have. I think it can be also very effective, but for us, we chose to use EEG in part because of the cost, because it is a significantly cheaper than most FNIRS systems at the moment. I think FNIRS would also be great, though, because it is somewhat resistant to movement. And in the end, we'll use whatever tool is available and that a person could put on by themselves. So like you saw in our home-based version, we just have the EMG for them because they can't put on an EEG headset on their own at home, especially if they only have control of one hand. And I think people are working on EEG headsets that could be put on with just one hand. So that's kind of why we went in that direction. But anything that reads out brain signals that is relatively robust and accessible, we're happy to try.
Okay, so Paul has two questions. So the first one, I guess, so Paul, you have to correct me if I'm wrong. I guess it's related to the part on MRI. So this large database. And so whether you have methods, sensorimotor outcome and functional outcome.
Well, which measures? Yes. Yeah, that's great. I did not pause to address that. First, impairment, we used Fugl-Myers, and for functional outcomes, we used a combination of the Action Research Arm Test, or ARAT, and the Wolf Motor Function Test, which are two kind of functional measures.
Okay, thank you. Sorry, can I interrupt?
Sure.
Etienne, apologies. What I may have misled.
It's good that you mix. Yeah.
Sorry. What I actually meant was you said that you'd taken some sensorimotor measures. I was thinking, in addition to the functional ones, did you measure things like neurophysiological measures, like evoked potentials and things? That's what I was actually asking.
Oh, I see. That's a great question. So each research group that contributed data measured a ton of things. So they might have measured, for instance, like motor evoked potentials, grip strength, physiology, things like that. Because we're trying to harmonize across all these research groups, we just took things that are relatively robust and easy to combine. So that's why we use mostly clinical outcomes. But we do have that data if you're interested.
Okay.
There's data to be answered.
Okay, Etienne, am I allowed a part B question, please, Etienne?
Yeah, so but you are here to tell it. But the question is whether you have looked at changes in basal ganglia volume over time.
Yes. Yeah, that's also a great question. At the moment, we've just looked cross-sectionally. That said, again, a lot of the data sets are longitudinal because people are doing studies where they do MRI, they do some sort of intervention, and then they do repeat MRI. So we do have quite a bit of longitudinal data. We just haven't pooled that data yet because it's very complicated since each group chose different time points, different start and end time points, and also different durations, and also different interventions in between. But it's something that we're very interested to look at because I think that tells you quite a bit more about the change and recovery, actually.
Okay, thanks very much. Great talk. Thank you.
Yes, thank you so much.
Questions, Your Honour.
Okay, so we had quite a few more questions if you're not too tired. So
Sure.
First, so I want to group two questions on your feedback, but first we take the other ones that is just how much money you do with your thing. No, whether you would say some VR on the market that would contain the rehabilitation game. Yeah.
Yes. Great. That's also a great question. We just got patent pending status for REINVENT. And there are a number of VR systems on the market. Actually, if you check out our Smart VR website, we have some talks that are coming up to discuss VR rehabilitation systems that are like commercially available already. We also, if you're interested, can partner with you to give you some of our like clinical games that are being used in rehabilitation as well. So those are kind of in a beta phase where we're just testing them and trying to make them better. But I think my guess is in the next 3 to 5 years, we'll see quite a bit more VR for rehab just because it's fun and something different that patients are a little bit more motivated to use and engage in.
Thank you. So yeah, a few questions about neurofeedbacks. That is, of course, very intriguing. So one first question is from Amina asking, putting cost aside, do you believe it's an effective tool?
Yes, great question. I think as with every therapy that we have, it just depends on the person. So if the patient is really motivated to use it and really likes the idea of neurofeedback, then it tends to be very effective. If the patient doesn't really care, they're forced to use it, for instance, it's not very effective. Part of that, I mean, I think I will be the first to say, like, part of it could be a placebo effect. I think placebo effects can be very strong and have real physiological changes. So if you believe that you're controlling your own brain activity, you might actually be able to change some things physiologically. But that said, I think there have been a lot of studies, and I think there was a recent review this past year or the year before, where there were case-controlled studies where people got control feedback, which was just sham feedback, that show that people who get real feedback do actually show better motor improvements after stroke. So I do think it works. I don't think it works for everyone, but I think you can know pretty quickly if it's going to work for someone or not.
Okay, so a question from Sophia also about MI neurofeedback. By the way, I'm not sure whether MI neurofeedback, I'm not sure whether this is your research or whether you were reporting as a research.
Oh.
Anyway, so she was asking, so which was the neurosignal used, for example, percentage of bolts change, and at which region of interest, etc.
Yes, great. That was my research. That's my postdoc research.
Oh, okay.
Yes.
So I know that, yeah.
Yeah. So we actually did it at the NIH, and I also got to do part of it in Tübingen, Germany, which was very fun as well. So I learned to do the experiment both in English and with a little bit of German. The regions that we used, we actually focused on real-time fMRI connectivity, so the connection between two regions. For individuals with stroke, we recorded from the primary motor cortex near the lesion and the thalamus, because we were looking at cortical subcortical connectivity, because that actually would be an advantage of using MRI, is that you get access to subcortical brain activity as compared to EEG or FNIRS, which are a little bit more cortically focused. And it was just looking at the correlation between the percent bold change in the two regions together. Okay.
So Sophia is also asking some, you mentioned that they were closing the eyes for motor imagery to get more concentration on motor imagery, and opens the eyes occasionally for seeing the feedback. So do you think this could confound the bold signal?
It's an interesting question. I think because it wasn't very regular and we were giving them real-time feedback, it should have, I don't think it would have made a huge impact on the bold signal. And I think also there would have been a little bit harder to track that even with eye tracking, to kind of know what they're looking at and when their eyes are open or closed, but it's a good consideration.
Okay, I have a question related to that. So, okay, so you presented so your result, your postdoc result on neurofeedback, MRI neurofeedback, and after you went to virtual reality with, if I understand well, EEG. So how do you go from one to the other? So which kind of VR? So is that really connected, or which kind of VR of feedback do you give to target brain areas or whatever?
Yes, yeah. So in terms of the feedback, so we could do it also on a computer screen and it wouldn't have been that much of a jump, but at that time when I made the transition to the EEG system, VR had also just become commercially available. So we felt that the more immersive system could lead to an embodied effect. And that's why we give feedback of a virtual limb, so like a virtual avatar versus like a thermometer or something that's less embodiable. So we actually do what we call like build a sensorimotor contingency with the person and the VR avatar they're shown. So when they first get in VR, we like take their hand and move it for them and show them that on the virtual screen it also moves. And so we say this is a representation of your virtual arm. And the idea there is that it actually activates regions that are associated with action observation and motor imagery. So like the premotor and parietal regions that are associated with kind of body representation. And that's also a part that's supposed to help.
Okay, so something totally different. Anybody is asking whether you are thinking of crowdsourcing the manual verification of lesion images in other ways than brain drive.
No, that was our only crowdsourcing attempt. We kind of felt like if it was going to take off, this is the best way, although we're very open to ideas or suggestions if they have other ways of doing it. I will say it took off at first. We got like a lot of people to use it, mostly like the parents and family members and friends of people in the lab. But it was not widely adopted. So we're not really sure how to get more people interested in using this. We occasionally get students in classes also to try it out and use it. But it turns out getting the public involved in science is a little bit more challenging, and there's probably some psychosocial elements that we're missing.
Okay. So Juan, who you have met before, is asking about the basal ganglia result. So you want to ask yourself?
Hi, Juan.
So
I guess I can, since you were reading. Oh, no, I forgot how I phrased my question. But yeah, I thought this thing about changes in putamen in after stroke were very interesting. And I'm new to stroke. Is there any evidence of, maybe this is naive or completely absurd, but is there any evidence of compensatory plasticity in the basal ganglia overall in the motor part of the cortical stroke?
That's actually, no, it's a great question. I think in order to have evidence of plasticity, you would need to have longitudinal measures, right, to be able to see change that, for instance, the non-injured putamen grows after stroke to a certain other region. That's something that we haven't explored yet, but we're very interested to look at. So I think at the moment, all we can really say is that if the volume of the putamen at this cross-sectional time point is smaller, then we can predict they have worse outcomes. But what I think you're pointing at is a much more interesting question and one that we hope to look at in the future.
Yeah, I also I was wondering if the basal ganglia are more or less plastic than cortex is, just because they are they come from different places in evolution in a way, and
Yes,
and
exactly.
maybe do you have any sense of whether there's a difference, or even if we know at all?
I don't have a good sense of if it would be more plastic than cortical regions, for instance. I think the rewiring, like the functional rewiring within the basal ganglia, could maybe be more plastic because there are a lot of, like, basically most cortical loops will go through the basal ganglia, or like motor control loops will involve the basal ganglia. So in that sense, there's a potential for it, but in terms of actual neuronal plasticity, I'm not as familiar. That's
Okay, cool.
a very good question. Thanks.
Oh, cool, thanks. We can chat a bit more later if you want that. Yeah, and thanks for the talk.
Sure, thanks.
Okay, so this is the question from Jinpei, I guess, about EEG. So the method you use, so do you record data and train a new motor imagery classification model for each patient, or do you train a channel model and perform calibration session on new patients? Yeah.
Yes. Also a great question. We calibrate to each patient. And actually, we calibrate to each session for each patient because we find that even over time, this can change. Even something as kind of like direct and straightforward as force output can change across sessions. So we do calibration each time.
Thank you. Yes, so really a lot of questions. So
thank you for the great questions. They were all very, very insightful.
Yeah. Okay. So Sophia is thanking you.
That's great.
So if we get to the end of first sets of questions in the chat line, I suggest we stop recording now and officially mark the end of the seminar, and Sook Lei for fantastic seminar. But we would like to suggest people who want to meet Sook Lei, camera on perhaps, and ask more questions to stick around, and because she can hang around a little bit while to meet you as well. So.
Yes.